Thank you so much, David. I'd like to begin by thanking David and CISA and the other organizers for giving me the opportunity to participate in this wonderful meeting again. It's always very enjoyable and instructive and stimulating, both in terms of the talks and the interactions and the beautiful places. So thank you very much for that. Happy to be here again. The title of my talk is Using Calorimetric Data to Drive Accuracy in Computer-Aided Drug Design. And just by way of background and context, the types of drugs that I'm focusing on here are often called small molecule drugs. Here are a couple of examples to differentiate them from drugs which happen to be proteins or perhaps nanoparticles. So a small molecule drug, for example, darunavir, which is a ligand and inhibitor of the enzyme HIV protease, an enzyme involved in the replication of HIV, is used to treat HIV/AIDS. Here's another example. Simvastatin, which is a drug used to treat high cholesterol levels, and it works by binding the human enzyme HMG-CoA reductase, which is involved in the synthesis of cholesterol. So a lot of small molecule drugs, most of them are work by binding to a specific protein that's involved in a disease process. And you'll hear me actually go back and forth between talking about these molecules as ligands versus small molecules. Ligand, just a term of art in the field, usually means a small molecule that binds a protein. The context, again, we're working in is usually one of structure-based drug design, where one has this three-dimensional structure of the target protein from the PDB, whether by X-ray crystallography or NMR. And here's an example of simvastatin with the active site highlighted in red, and you can see of HMG-CoA reductase with the active site highlighted in red with simvastatin bound there blocking the action of the enzyme. And so one of the key challenges in drug discovery projects where you have the structure is, okay, I have the structure of this protein, here's HIV protease, to figure out what compound to buy or make that will bind your target with high affinity and thus be a candidate drug. And I say candidate because, even if it binds your target, it still has to be safe and absorbable and so forth. So this is only one step in the process. But it's an important step. It's a step which is both costly and time-consuming. This is from an analysis by Bernard Munoz at Lilly from a number of years ago, looking at the cost in millions of dollars here of various steps in what's often called the drug discovery pipeline. And in orange are highlighted the steps that are involved in discovering a potent ligand, a small molecule that will bind your target protein with high affinity. And in blue are the subsequent steps, which are things like putting the compound into animals for initial toxicity testing, and then going into clinical trials. And I think many of you know that it's the clinical trials in a given project that are usually the most costly. But what Munoz pointed out is that a lot of compounds, a lot of candidate ligands that come here drop out early on. They're toxic to animals. They don't go through the clinical trials. And so you actually end up having to go through this ligand discovery process multiple times on average to come up with one drug that actually makes it to the market. And so if you then amortize these costs over all those efforts, the actual cost of coming up with a high affinity ligand, even before clinical trials, rivals that of the clinical trials themselves. And it also takes a good amount of time, which delays getting a medicine to patients and delays getting profit to the drug companies. So the vision, in a way, for computer-aided drug design in the context of structure-based drug design is to speed this process. The idea to get to high potent ligands quicker, do it at less cost, and also more cleanly, because if you don't have to do as much experimental trial and error, you don't waste as much, you don't flush as much chemicals down the drain. And I think there's also ultimately a prospect for making better drugs, because you can imagine that if we become really good at predicting that a compound will bind a given target, we could then say, well, what other human proteins in the PDB, for example, is this compound predicted to bind? And can I use that to design compounds that don't have off-target effects that would produce side effects? Or, if we get very clever, could we come up with compounds that would have significant activity against multiple proteins in a given pathway and thus be more effective at shutting down the pathway with only one compound? And ultimately, in a world where we're all going to be sequenced at some point, and if people get cancer, the genomes are sequenced, there's the possibility of using this kind of technology to help select a compound that would be most effective against for a given individual based on genomic information. But we're not there, okay? And this is really a lot of what my work focuses on, is how to, we make progress towards this vision. And so what I want to focus on is a challenge of computing the binding affinity of a small molecule for a protein. So the scenario is we have a candidate ligand with a protein that's targeted, and we want to estimate the binding free energy, the binding affinity. Usually this occurs by non-covalent forces like the hydrophobic effect, Lennard-Jones dispersion interactions, and hydrogen bonding. Some drugs do bind covalently, but I won't be focusing on those here. And the theory, the sort of theoretical framework for doing this with statistical thermodynamics is something I published in 1997, is summarized here. This is the binding free energy, and the sort of heart of it is this ratio of configuration integrals, which I won't go into, but you can see that it's a ratio of a configuration integral for the complex, this is the PL subscript, versus the protein and the ligand. And intuitively, the configuration integral is an integral of the Boltzmann factor over all conformations. The Boltzmann factor is large for conformations of low energy, so the more low energy conformations that this complex or protein or ligand can adopt, the bigger the configuration integral. So if this ratio is large, we get a negative free energy of binding. And this statistical mechanical framework has been used to and can be used to construct or at least rationalize a whole array of computational methods for estimating protein ligand binding free energies, from relatively simple and fast docking and scoring methods that I'll touch on in a moment, to more detailed methods that are often called explicit solvent free energy methods that I'll also talk about, that are sort of at the other end of the spectrum of complexity. But one thing that you'll notice that all of these methods require is some estimate of the potential function, this U, as a function of conformation. These potential functions are also often called force fields. And basically what they tell you is for a given configuration of the system, where all the atoms are in space, what is the potential energy? And they typically have terms that account for bond stretches, torsional rotations, van der Waals interactions. There were often point charges on atoms to account for interactions among charge groups and hydrogen bonding and so forth. And of course, there are also a range of sophistication of functional forms, but today I'm going to be focusing on the sort of canonical functional form, which is widely used in fast simulation codes. And this is just an example because I'll be talking in a little bit about water models. This is a TIP3P water model, TIP4PEW, both essentially work from Bill Jorgensen, pioneering work, still used. And these show the partial charges on the oxygen and the hydrogen in these two different models. So now back to the method. So I'm going to sort of give you the two ends of the spectrum. So docking and scoring is basically a technology where you take the structure of the protein, stick the candidate ligand inside, do some sort of conformational search, position, orientation, and look for the conformation of lowest energy. And then typically the interaction energy of a ligand with a protein is read out as some sort of estimate of the binding affinity. There are a lot of codes that do this. This work goes back decades, it's a fairly mature technology, arguably. So from the way I described it, you can see that there are some approximations in these methods. One of them is that the proteins are typically held fairly rigid, and as highlighted beautifully in the last talk and others, proteins are not rigid. They typically use a rather crude treatment of water, which we're going to move away from in the next class of models. They often neglect configurational entropy, the entropy changes associated with the motions of the ligand and the protein, or they treat it very approximately. And they also, one of the big problems is getting the protonation states right, particularly for things like histidines, where the pKa is so close to neutral that it's hard to predict what the protonation state will be and whether indeed it may change on binding. And again, as I touched on before, the force fields have approximations, and this is going to be important in my talk. So at the other end of the spectrum of complexity, arguably you can use molecular dynamic simulation. So here you take your potential function. Usually there's a lot of explicit water swimming around, maybe you have some sodium and chloride, phosphate, whatever your buffer is, and you set the whole system into motion. You basically couple it to a heat bath by appropriate methods, and you solve Newton's equations of motion while allowing for coupling with an environment at a certain temperature and pressure. And if that's done correctly, what you end up with is an ensemble or a trajectory of configurations that are drawn from the Boltzmann distribution corresponding to your potential function. And those Boltzmann distributions can be used to compute thermodynamic quantities. So here's one approach that's fairly widely used in the pharmaceutical industry today. It's called relative binding free energy calculations using an alchemical method. The reason it's called alchemical is because it involves non-physical pathways that I'll describe. The basic method for all these free energy calculations is you're not typically computing configuration integrals directly. You're computing the reversible work connecting two states. And then you use the fact that the reversible work is the difference in free energy. So here, let's say there were two candidate ligands we're interested in. They both could bind this protein. We want to know what these two binding free energies are. You can actually sometimes save computer time by saying, well, what I really want to know is which one's better. So I'm perfectly happy with the relative binding free energies. And what was shown in really pioneering early work from Tembe and McCammon in 1984 is you can play a trick on the computer that you can't do in the lab. You can take this compound, add the group that makes them different in solution and compute that reversible work to get that non-physical free energy difference. And you can do the same thing in the binding site. And since you now have a closed thermodynamic cycle, the fact is that I'm not going to get my signs right, but the difference between these two equals the difference between these two, which is what you really wanted. So it's a trick to make the calculation simpler because it's often, it can be difficult to compute the work of actually pulling the ligand out of the binding site, although we'll be doing that later. So these methods capture more detail, arguably, than docking and scoring. They allow the protein to move. Remember, everything's wiggling and jiggling with thermal energy. The ligand is moving. But they also have sources of error. And probably the three main ones are shown here. One of them is that, especially for proteins, which are complicated and have motions across multiple timescales, it's hard to be confident that the work you're calculating is the reversible work that the force field is trying to tell you. The calculations may not be converged. Another way of putting it is that the calculations are numerically imprecise. In addition, again, we can have issues of the signing protonation and tautomer states, and again, the accuracy of the whole thing is dependent on the quality of your force field. So we've had this array of methods that have been developed over many years. Computers have gotten faster and faster. This is sort of a classic Moore's law graph of computers' speed in instructions per second on a logarithmic scale versus year and decades. And we have this fantastic increase in computer power. And the question is, okay, have we benefited? Have calculations gotten more accurate? This is some measure of accuracy of your calculations as a function of year. And it's actually rather hard to tell. And one of the reasons, there are probably two main reasons, I would say. One of them is that when people develop new methods, they typically test them on systems where they already know the answers. So they go into the literature, pull out some data, do my calculations, oh, it agrees, it doesn't agree, whatever. And the problem with that is that it's all too easy, and I'm not trying to accept myself from this, but it's all too easy to introduce unintentional bias and sort of do some tuning that makes things work. So it's hard to know whether the method will work as well when used in real life. The other problem is that different methods are tested on different systems. So if I develop a method and test it on systems A and B, you develop a method and test it on systems C and D, and you do better, it might be that your method is better, or it might be that systems C and D are easier. So in order to develop the possibility of having rigorous objective evaluation of these computational methods, I've been working with Romy Amaro, another professor at UC San Diego, and also Stephen Burley at the PDB, to hold what are called blinded prediction challenges. So some of you will have run into these. The basic idea is that we collect hitherto unpublished protein ligand affinity data from the drug companies usually, because they're the ones who don't publish, right? If we develop data, we usually try to publish it. They hold on to a lot of it. But as projects get older, patents are settled or whatever, some of the data they're willing to release to us. They'll release data to us. They'll tell us what the protein is, what the compounds are, and what the affinities are, whatever measure of affinity. And we will release that information without the affinities to the computational chemistry community with an invitation to participate, make these predictions. And then we release the actual data, and there's cheers and gnashing of teeth depending on how people did, and we hold a workshop and discuss what we can learn. And hopefully it's a method of both objectively assessing methods and also of ultimately driving progress in the field. And having participated in these before running them, it's definitely an instructive exercise. So what I want to do now is tell you about the selected results, which are fairly representative of other results we've gotten. I'm just going to pick one case. This is from what we call Grand Challenge 2, which was held a little over a year ago. The target protein, don't worry about exactly what it is, but it's a well-behaved protein, farnesoid X receptor. There were data that were kindly provided by Roche, and the data that we're focusing on here are something like 102 IC50s, which we think are a pretty good stand-in for, at least for getting relative binding free energies. And from those 100, we actually pulled out what we called free energy subsets. So these are subsets of compounds that we thought would be amenable to these alchemical methods I showed you before. What that means is that if you want that alchemical method to work for you, you don't want the compounds to be too different. The point is it really works well for compounds that you're sort of doing localized SAR, where you're modifying one group at a time. And so, for example, here's a subset of compounds. Here's a starting compound. You can easily turn it into this one by adding this chemical group. You can modify this substituent, or you could just delete it. So this is just an example of the types of differences among the compounds within each free energy set. And so these are results from that challenge. And on these graphs, what you're seeing, this is a result for one free energy set, and this is for a separate one. Each column is the submission from one group. So this is how well each group did. And the measure of error is root mean square deviation of the computed versus the experimental relative binding free energies within the set of compounds. And what I've done is to color the submissions according to the methods used. So in red are the ones that use the more detailed free energy simulation methods, and in blue are the other ones. So these include methods like docking, but also methods that I didn't talk about that are sort of intermediate complexity. And some of them also, I would say, have been trained on existing data for similar systems. But what you see is that in the red ones, which are the free energy methods, which in principle capture more physics, are not doing any better. In this free energy set, they tended to do worse. Here, their performance is scattered everywhere throughout. So one of them was the best, although there were others that are within the error bars, and others were not so good. So, this was kind of interesting. Why is it that, and sort of contrary to expectations, that these more detailed methods would not outperform the simpler ones? But actually, so let's go back now. This is from a paper from the company Schrödinger, which has really been pushing this technology. This is sort of a landmark paper they put out, a retrospective study now of something like 200 protein small molecule binding affinities that they were computing by these methods. And this is a scatter plot of the computed relative binding free energies versus the measured experimental binding free energies. And you can see that there is certainly a correlation, so something is certainly going right here. But there's also a fair amount of scatter, which I think is sort of consistent with these kinds of results. Maybe a little bit more provocatively, these are data which I pulled out of a table from a paper of theirs about 10 years ago showing results of their latest docking and scoring method for about 200 protein ligand systems. And so even if you look at their own data, again, I'm maybe pushing this a little bit, but I think this is what the data show. It's pretty similar, docking and scoring and the free energy method. So the question then is, okay, if we have these free energy methods and they're supposed to be better, they're capturing more physics, what's going wrong? And so we go back, and I guess what I would say is that in principle, they're capturing all the detail of this, or in principle, full implementation of this theory, modulo the protonation state. So what could be going wrong? We talked about that before. One possibility is that the simulations are just too short, and maybe you have to run longer for that protein to undergo that conformational shift, and suddenly the free energy will correct itself. Another possibility, and this can be challenging to be sure about whether you've run long enough. Another possibility, again, is the protonation and tautomer states. And the third possibility is that the force field is not doing it. So what we've been doing for several years now is going back to simple model systems for binding. So instead of looking at proteins, we look at host-guest systems. So as many of you know, a host, this is different terminology, but it's like receptor ligand. So the host hosts a small molecule that's called a guest. Host molecules usually have a cavity or a cleft. This is a cucurbitural with a hole in the middle. This is a cyclodextrin. You can see there's a hole in there. And what's nice about these systems is they bind small molecules from aqueous solution, just like proteins do. But because they're so simple, it's easy to run simulations that are long enough that you can have much more confidence that the results are converged. So we can largely eliminate the convergence question and get results that are numerically precise. We can also choose systems where the problem of protonation and tautomer states are avoided. So maybe if you're going to have an imidazole, then don't do your experiment near pH 7, for example. And so we've eliminated these two problems, or we can, in principle, eliminate those. And so now, if we do careful comparisons with the experiment, we should be testing the force field itself. So let me show you a little bit how we do these comparisons and some of the results we've obtained. So we call this computational calorimetry. I guess what really made it a little bit beyond what people have done before is that we found that for the host-guest systems, which are fairly manageable computationally, we can really get precise, numerically precise calculations of binding enthalpies as well as free energies. And the point here is the high precision in terms of the results, that the simulations are run long enough that we have confidence we're getting the results that the force fields are trying to tell us to within a few tenths of a kcal per mole. So I talked about the alchemical binding free energy methods. We actually use what's called an absolute binding free energy method that I call attach, pull, release. And it's diagrammed here. So let's say this is your host, or it could be a protein. This is your guest or your ligand. So we start off by running a simulation with all the water in the bound state, just like this. And we compute the work of gradually and reversibly attaching a spring between the two molecules, just so I can get a handle on them. And then we use that as a handle. We increase the equilibrium length of the spring to push the guest out of the host. And although I've shown it as being done in one jump, it's done in a series of static windows. So we essentially get the force at each stage, and you can integrate force over distance to get a work. And then we compute the work of releasing the spring. This is all done while running MD at a well-defined temperature and pressure. So essentially, we get the reversible work of going from the unrestrained bound state to the unrestrained free state, which should be the binding free energy. For the binding enthalpies, we found that it's pretty simple to do. We run a calculation of the host-guest complex where they're held apart by an artificial spring just to keep them from binding with a bunch of water and the solvent, whatever experimental conditions you're trying to model. Compute the average energy, just the average energy given by the force field over all the snapshots in the trajectory. And then we do the same thing where we've taken away the spring, and we do it for the bound state. And we take the difference in the average energy. And that's our result for the binding enthalpy. The delta PV term, of course, which you're aware of, is trivial because the volume changes in these reactions are trivial, and 1 atmosphere is not a high energy density. So this is the calculation. So let me show you how some of these calculations have gone. So this is cucurbituril, which is this circular host molecule in water binding to a bunch of guest molecules. But cucurbituril likes to bind hydrophobic clumps, chunks, with a polar part, especially a cationic part. The cation tends to sit near these carbonyl groups at the rim. And the data, I don't know if I have the data reference. I think some of the data from here, but I think some of them are from Lyle Isaacs' work. So that's cucurbituril. Here's a panel of alpha and beta cyclodextrin. Those are, as you know, rings of sugar molecules that form a little cup, as shown here. We did calculations for 24 carboxylates, 11 alcohols, and 10 amines. So we have a range of charges for the experimental conditions at neutral pH. And there's experimental data from all of these that were actually from this compilation. So I'm not going to spend a lot of time on the computational details. Suffice it to say that we do fairly long simulations, about 1 microsecond for the enthalpies, and about 50 nanoseconds per window as we pull the guest out. And we use rather sort of vanilla force fields, the kinds of force fields that are typically used in protein ligand computer-aided drug design modeling. And I would say that the real reason why we can do this, one reason why this hadn't been done before, is that GPUs make computing so much faster now. You can do these microsecond calculations in a day or two. So let me show you some sample results. So this is results for the cucurbituril with these guests that I showed you before. This is going to be a little summary. The force field, we're using something called general amber force field for the bonded terms and Leonard-Jones terms. Again, this is a rather standard force field in the field. We're using this TIP3P water model I showed you before, which is also widely used. And we're getting the partial charges from a rather fast but widely used method called AM1BCC. This is the statistics for delta G and for delta H. What you see is that the correlations of calculation with experiment are good. This is the square of the Pearson's R. But the root mean square deviations and the mean signed errors are pretty bad. So there's a big shift. Basically, we're grossly overestimating the binding free energies. So let's try the TIP4PEW water model, and the reason you want to try that is that it gives somewhat better properties for pure water. So it seems like it should give better results in general, but it doesn't. It gives similar correlations, but actually worse overestimates of binding free energy, which is pretty interesting. So I think one of the messages that I take home from all these studies is that optimizing a water model for bulk properties doesn't necessarily tell you it's going to work well for other problems of interest. And there may be surprisingly large differences in the results for water models that are really very similar to each other. So now going ahead and saying, well, let's go back to TIP3P and use a slightly more sophisticated set of charges, which are known as RESP, which I won't go into here, but they're slightly better regarded in the field. What we find is the correlations get a little bit worse, but what's nice is that the mean signed errors drop, so we're not overestimating the binding free energies or enthalpies as badly. But there's still a good amount of scatter. So things, I think we learned some things, but one of the things we learned is that the force fields are not quite doing it for us here, which is consistent with what we saw for the proteins. So this is a summary of data for the cyclodextrin host with the 40-odd guests of various charges. This is experimental binding free energy versus computed. Here's a scatter plot. We have both experimental and numerical and computational error bars here. And you can see that there's definitely correlation, but there's a fair amount of scatter. This looks a lot like what we saw for the protein ligand data, actually. One thing that was sort of encouraging here is that if we take exactly this graph and just color the points now by the type, so the amines are blue, the alcohols are green, and the carboxylates are orange, there's definitely, different, each one has a different bias. And so this perhaps is a hint that by rejiggering the force fields for these different functional groups, we might be able to bring these into alignment and reduce the scatter. These are the results for the binding enthalpies. I would argue these results are worse as a larger systematic error, and if we color the points by type, there's no longer any obvious direction for improving things, but at least it gives you a sense for the comparison of calculation with experiment. And so that's work from our lab, but we've also been involved in holding blinded prediction challenges for host-guest systems. So just like the protein ligand, we do it for the host-guest. And this is similar data to what I showed you before for protein ligand free energy predictions, but for different sets of host-guest complexes. This is something called octa-acid from Bruce Gibbs' lab in Tulane that he's synthesized and characterized and generously donated data. And this is data from Lyle Isaac's lab at University of Maryland, who's also been a great donor of data for these challenges. And you can see different submissions. These are not too bad, but for this one seemed to be harder. The errors are larger. So there are significant errors in these methods, not only from our group, but from other groups as well. And so essentially what I would conclude from these results is that by using these host-guest systems as models, we can confirm that there are significant and pervasive problems with the force fields that are typically used in computer-aided drug design, confirmed from our work and also by blinded prediction challenges. And although I didn't go into it, some of you may be familiar with a more sophisticated force field called amoeba from Wren and Ponder and others. And even when they add more things like electronic polarizability and multiples and fixed multiples and so forth, it's not clear that they're doing any better. And so I think it's fairly clear that these force fields form the basis for a whole array of CAD methods, computer-aided drug design methods, and if we don't do better with them, we're not going to make a lot of progress on accuracy. So why is this the case? Why are the force fields problematic? And there's a bunch of different aspects, but what I want to focus on is that the types of data that have been used to parameterize them. So there's the main types of experimental data have been pure liquid properties, things like densities, heats of vaporization, and so forth of liquid benzene, liquid acetone, and so forth, trying to get those right. Some use of small molecule hydration data, so the free energy of moving, let's say, ethanol from gas phase into water. And for proteins, some use of conformational preferences from NMR studies and so forth. That's not really relevant for the host guest. And I think the problems here, the limitations here are several-fold. One is that the number of data that people are using at this point is smaller than the number of parameters, so that's obviously a problem. In my view, another problem is that if we're interested in binding calculations, we probably should try to use binding data to do the parameterization. On one hand, you could say, well, then we're limiting transferability of the force field if you're tuning for one type of observable. But right now, we're tuning for these observables and not getting very good results for binding. So I feel as though adding more relevant data, more and more relevant data would be useful, and particularly because I think binding calculations, binding data, probe the force fields in different ways. And you saw that with the water model. When we went from tip 3P to tip 4P EW, and from a not quite so good to a better water model, the binding results got worse. Now, who would have known that? And I think what that may reflect is things like the fact that when you have binding, you have water in a protein binding site, the water structures up. It has, different thermodynamics, if you will, than bulk water. And if you're not taking account of that in tuning your water model, there's a pretty good chance that the binding calculations won't work out, because then that water gets displaced, goes back to bulk. So those differences matter. In addition, since most of the parameterization has been based on pure liquids, it limits the amount of chemical interactions that we're probing. So you may have acetone-acetone interactions or benzene-benzene interactions, but what about the mixture? So I think that's an argument for using liquid mixtures, and also for the binding calculations, which put different types of groups together. Now, you can use quantum calculations to parameterize a force field, and we do. We as the field do. The benefits are that they're pretty straightforward to generate. You don't need to buy chemicals. You can just do some calculations. The accuracy can be high for what they're giving you, and they can probe virtually any interaction type that you want. The drawbacks are that without incredible care, and there's a couple of examples of this, but it's not easy to map calculations from the gas phase to the condensed phase, because things change when you get into the condensed phase, partly because of electronic polarizability, partly because other things change as well. So what we've been interested in doing, and I'm going to show you one example of this, is actually using binding data to parameterize force fields. And the basic idea is, if we have an initial set of force field parameters, we use some simulations to compute experimental observables. We then compare the calculated and experimental results. And here, in addition to liquid properties and so the things I talked about before, we want to add in host-guest data. We then use some method to adjust the force field parameters, try it again, and iterate until hopefully we have a better force field. So what I want to do now is show you a first proof of principle for doing this. This is a study where we used, I think, 6 data to do the parameterization, but we then tested on something like 120 host-guest data. And that number is already pushing into the order of magnitude of liquid state data that people typically use for parameterizing and testing force fields. So here's our training set. This is that octaacid I showed you before. It's basically a little hydrophobic cup that is water soluble because of some acidic groups, again from Bruce Gibbs' lab, and here are some gas molecules. And what we did is to compute the binding, we only used the binding free energies in this case, for guest 1 through guest 6. And I don't expect you to go deep into these numbers, but let me tell you what they are. This is it turns out you can write code to extract from the simulations the partial derivative of the computed binding affinity with respect to the Leonard-Jones parameters of your water model. You could do it for any other parameters in the system also. Here we were just looking at the water model. So by getting these partial derivatives of the computed results with respect to force field parameters, that lets us set a gradient that lets us choose a direction to adjust those parameters that will bring the calculations in line with experiment, at least to, better in agreement. And you can actually then make a little contour plot, essentially of error with respect to, in this case, the sigma and epsilon parameters of the water model. So I should have said we're starting with the tip 3P water model, which is very simple, and we're just trying to adjust 2 parameters. It's a proof of principle, hopefully. This is a contour plot. You can see in yellow is where the greatest accuracy is, the lowest root mean square error of calculation versus experiment, and it gets worse on out from there. This is where the initial calculations were. Now, we would in a way like to go over here and get the lowest error possible, but we found that when we did that, we got a water model that didn't give good bulk water properties. And I don't think we should make those absolute requirement, but I don't want to have a water model that gives crazy water properties. So we ended up sort of compromising and going over here in this new model called bind 3P, which is really very similar to tip 3P. Very simple adjustment. We kept the charges the same, and just did these small adjustments on the Leonard-Jones parameters. So this is essentially the diameter, twice the radius of the water, and this is the well depth for the van der Waals interaction. These are the pure water properties. This is experiment. This is the original TIP3P model. This is the bind 3P model. And you can see that the density is okay. The dielectric constant it was too high and it's still just a little bit higher now. The isothermal compressibility is maybe a little bit better. The coefficient of thermal expansion, well, we got it wrong before and we still get it wrong. And the heat of vaporization is okay. So we haven't screwed up the bulk water properties. One thing I'm not showing you is the radial distribution function has a lower first, maybe second peak, but the water structure isn't much worse than that of TIP3P. So what I'm going to do now is take you fairly quickly through a summary of data. I told you that we tested this on something like 120 test cases, which were not used for the parameterization. Long story short, everything gets a little bit better. Not dramatically better, but a little bit better. So this is octacid again, the same one we trained on with a bunch of additional guests, and we'll look at free energies first. And I don't want you to worry about all this, but these are the different guests. This is experiment, this is the experimental method, this is the new model, this is the starting model. These are some other water models that we also put in to check, but these are the summary data at the bottom. So by going from TIP3P to bind 3P, the correlation remained about the same, the root mean square error came down, and the tendency for overbinding was diminished. So we reduced the bias. And that was true, actually it's better than these other water models also for these particular cases. Now we look at enthalpies for a nearly overlapping set of guests. We just used whatever was available experimentally. And here again, going from TIP3P to bind 3P, the overestimation gets less bad. This is the mean signed error. The RMSD goes down. The scatter was the correlation was pretty bad to begin with, and it didn't get better or worse. There's one other water model that's done better, which is actually a pretty good water model, but it doesn't do so well in the other tests. Now this is a slightly different host molecule with another set of guests, also from Bruce Gibb. Again, in summary, we didn't do all the other water models here, but going from TIP3P to bind 3P reduces the overall error without messing up the correlation, at least not very much. This is now also with enthalpies. And then this is the larger test set. This is something like 69 guests. There were, with both free energies and enthalpies for cyclodextrins and some cucurbiturils. So here the cyclodextrins, delta G, delta H, we didn't have enthalpy data here. You can see going from TIP3P to bind 3P reduces the tendency for overbinding, not hugely, but it's in the right direction. All we did was tune two parameters. The correlation stays about the same, and you see sort of similar results across the board. So just to summarize this bit of the talk about this bind 3P water model, I think it's a useful water model. I think if I'm going to be doing these calculations in the future for design purposes, I'll use bind 3P. But I think more significant is that we've actually shown you can use host guest binding data to tune force field parameters for the first time, and that you can get significantly improved results across a panel of test cases that weren't in the training set. Didn't improve the scatter, but it reduced the bias. So what that does is sort of lay the foundation for the work we're now starting, which is to address the limitation of the host guest data set that's available in the literature, which is that there isn't a huge amount of chemical diversity. There's a ton of data for cyclodextrins because you can buy them by the kilogram for cheap. And there's a few others where that are not too hard to synthesize, but there isn't a whole lot of chemical diversity. So what we've started doing is making our own. So the idea is, if this is cyclodextrin, I'd like to be able to add, in sort of a modular fashion, a variety of different chemical substituents on one or the other rim. And we're focusing on this, what's called the secondary rim. That's where the secondary hydroxyls are. It's a wider mouth because we think the guests are going to stick out there more and have more opportunity to interact with the groups we're adding. The idea is to have a bunch of cyclodextrin derivatives that we can run across this axis of a matrix, do calorimetry measurements of them with a bunch of different guests that we can run down this axis, and build out a new matrix of experimental data of protein ligand binding that could be used to test and parameterize force fields. So to do this, we needed to build up our synthetic capabilities, which started at zero, so it was easy to. Katie Kellett, who is doing host guest work in the UK and has joined my lab. And she's solved what has traditionally been a rather hard problem of adding substituents to the secondary rim of cyclodextrins. She's got ways of adding them now to the 3 position. And we'll be also looking at the primary hydroxyls later just to see whether one face is more interesting than the other for these derivatives. But so she's developed syntheses for diverse mono 3 substituted cyclodextrins using amidurea and thiourea linkages. And what's nice about these for lazy people like us is that they're one-pot syntheses with trivial purifications just by precipitation with yields on the order of 50 to 80%. So I don't know if this is visible, but each of these little things in brackets is just sort of the rest of the cyclodextrin. And you can see the side chain sticking up here. So here are some carboxylic acids, here are some aromatics, here an amine, there's some amino acids. This is actually glutathione at 58% yield. This is a furan. And actually, there's somewhere in here there should be a beta amino acid. Actually, the trick to this is largely avoiding the steric problems. Turns out you can pretty easily add beta amino acids on here to generate sort of biologically relevant diversity. So this is in some ways in concert with a larger effort, what we call the Open Force Field Group, which brings together a number of labs. So John Kodera at Sloan Kettering, Dave Mobley at University of California, Irvine, Michael Schertz at University of Colorado Boulder, Liping Wang at University of California, Davis, as well as Chris Bailey, who's done sort of widely used force field development over the years, and Ken Kronlein, who's here, who's helping us a lot with understanding the data and finding the right data, high quality data to use. So I'll summarize. I've provided evidence that even though they're more detailed and sophisticated, these free energy methods are not clearly more accurate than faster competing methods. We've used this computational calorimetry approach to provide evidence from calculations on host-guest systems that at least a big part of the problem is the force field. I mean, they're still predicting protonation states and so forth, and convergence. I think we've shown the first use of a body of host guest data to reparameterize a force field, at least to a small extent, and then to test. And we've shown a new synthetic access to varied cyclodextrin derivatives that we think will be useful as we build out this process. So many people have contributed to this work. The D3R project is done with Romy Amaro and Steve Burley as PIs, and Vicky Thayer, and a number of students, postdocs, and staff. The host guest calculations and experiment combine staff and synthetic folks and computational people, and the open force field effort also involves a number of labs, and this has been supported essentially by NIGMS. So thank you very much for your attention.